The overall goal of this procedure is to create chimeric embryos by transplanting ectoderm from donor embryos to the developing upper jaw of chick embryos. This is accomplished by first removing the donor ectoderm for transplantation. Then, the host site is prepared for engraftment. The final step of the procedure is to place the donor tissue on the host site and affix the graft in position. Ultimately, results can be obtained that show exclusive engraftment of donor ectoderm, and through molecular and morphological analysis, the function of donor ectoderm can be determined.
This method can help answer key questions in developmental biology, such as identifying the role of various tissues during morphogenesis. Demonstrating the procedure will be Dr. Diane, a senior investigator from our laboratory.
Before beginning this procedure, prepare media and ensure all required solutions are accessible and at the correct temperature. Then, whilst wearing adequate eye protection, pull microcapillary tubes to a fine point over an alcohol flame to make glass pins. Sharpen tungsten needles by holding them in the flame of a propane torch. Begin extraction of the donor embryo from the shell by placing a piece of tape over the end of the eggshell. Then, use the point of a dissection needle to create a small hole in the center of the tape. Insert a 10 ml syringe and an 18 gauge hypodermic needle into the hole and remove 1 ml of albumin from the egg. Then, using a dissection needle, make a small hole on the top of the shell. Place a second piece of tape to cover this hole. Cut a circular opening in the egg. Using 2 pairs of forceps, grasp the embryo and place